So just to add to what Julia said, so I'm an exploration minerals and soils geologist. I've worked extensively over the UK for the last 35 years, looking for all kinds of minerals, primarily aggregates and horticultural sands, dimension stones, and set up Rhizophilia 2 years ago because the story, to elaborate on what Merlin says, is just too great a secret to keep to yourself. Having spent more time in fields, hundreds and hundreds of farmlands across the UK and around the world getting sprayed with glyphosate, hit by nitram fertilizers. And when you stand in a field, 1.5 miles by 1.5 miles of potatoes that are wilted off through sulfuric acid, and the soil even feels like concrete, and it's silent. You are living Rachel Carson's Silent Spring. Imagine, if you will, that the distance between these two beautiful flint pebbles, sorry, geologist in me, is representing the age of the Earth, okay? This beautiful Rudbeckia represents land plants. Land plants came in about here in that timeline. The reason I'm using Rudbeckia to represent land plants, very important reason, I just love Rudbeckia. Isn't that just sunlight made physical? Angiosperms actually come about here in my timeline. 470 million years ago, during the middle of the Ordovician period. And since then, they've been working with the soil food web microorganisms to create a flourishing folial fantasia on planet Earth. When I talk about the soil food web, I'm talking about the bacteria, the fungi, the protozoans, nematodes, and microarthropods. And when I talk about working with the soil food microorganisms, what I mean is actually being enabled by the soil food web microorganisms who frankly invented plants. Let me explain. Hang on a minute. Very important this bit, bear with me. Okay, there's no oxygen in the atmosphere beyond this point. Thank you. Okay, let's get through this, no disturbance, please. So, age of the Earth. Just beyond that door, I could go, bang! Okay, right. Age of the Earth. Bacteria come into the picture about here. Archaea, our ancestors, ancestor of all eukaryotes, including fungi, come in to the story about here. About here, we've got protozoans, aquatic fungi, land fungi. About here, we've got nematodes, and then literally just before plants evolved, we've got the microarthropods. Between these guys, they invented every primary and secondary metabolite and every metabolic process essential for terrestrial life on Earth. They created photosynthesis, glycolysis, citric acid cycle, oxidative phosphorylation. That's the last stage in respiration that you're all doing as I speak. Vitamins, B12, they can produce that, we can't. Enzymes, we can produce about 17 enzymes in our body. Get in. One bacterium, Bacillus subtilis, can produce over 300 enzymes for metabolic processes. Aminocyclopropane carboxylate deaminase. Ha! Easy for me to say. I'll come back to that one shortly. I better put a slide up, hadn't I? Oh, that's it, sorry, I beg your pardon. The beginning. In your picture it says the beginning. When I wrote beginning, or beginning, it actually auto-corrected it to beginning. It should be beginning. That's where it all started. A muddle and the endophytes. Okay. But those processes also included horizontal gene transfer, one of the most extraordinary processes in life. 580 million years ago, about here, a bacteria, a cyanobacteria called Nostoc, shared some genes with streptophyte algae that enabled that algae, as Merlin alluded to survive desiccation when the ponds dried up. I wrote a little poem for my Hampton Court show feature. Anyone want a little bonus poem? Bacteria and algae living side by side. Pond dries out. Poor algae dies. Bacteria says to algae, please don't hesitate. Take these genes and you won't desiccate. Algae says to bacteria, that's a pretty cool trait. Let's stick together and proliferate. Thank you. I'm here all week. Thank you very much indeed. Thank you very much. Thank you very much. 580 million years ago, over 580 gene families were transferred from bacteria to the streptophyte algae. The vast majority of them were metabolic processes for surviving life on land. So important, those genes were conserved in all seed plants. Now, there isn't much mineral in seawater. There's a lot, but not enough if you really want to colonize the land. So roll forward 100 million years, and fungi come into the picture, and fungi start to share genes. They share genes with insecticidal properties. That gene above the red dot, FB-lectin, KP4. KP4 stands for fungal killer gene. And the fungi enabled the plants to mine nutrients. They had the wealth of the Earth's crust beneath them. That enabled plants not just to survive on land, but to thrive on land. Now they could actually grow much greater biomass because of the minerals in the soil. Well, rather, there wasn't any soil at the time, but mineral in the proto-soil. If land plants are the answer to the question, how can we microbes spread our influence into the airspace above solid rock, thereby enhancing our abilities to regulate the planet for the benefit of all organisms, then this bit between there and there is the working out. Do you remember at school, in your exams, you'd get 1 point for stating an answer, but you'd get 5 points for the working out? Yeah? This is the bit that we as gardeners and growers, and society in general, at the very best, take for granted, and at worst, don't really know about. This is the bit that we should be thinking about, what these guys are actually doing. The result of that colonization of land were the embryophytes, the liverworts, the bryophytes, and the mosses. I should carry this thing with me, shouldn't I? I just like my stick. That's all. Bonus slide. Bonus slide. Ever thought of what the land looked like when, or the earth looked like when plants actually colonized land? Here we go, 470 million years ago. That's Tunisia, right on the South Pole. You can see the shape of Africa there. There's Britain, there, on a little subcontinent called Avalonia. You notice Scotland is missing. Scotland, I should have brought a bigger stick, is there, on the southern part of the North American continent. 30 million years prior, that little slice of land was joined to Florida and North Africa. And then it split away in a volcanic island arc, heading for a collision course with Laurentia. That's what the Earth looked like when land plants began their journey. The muddle in my title. So successful were the embryophytes at colonizing the land. They dragged so much carbon dioxide out of the atmosphere that they helped create a snowball Earth, a runaway global cooling. At the end of the Ordovician period, 443 million years ago, it happened again. The fungi, after this event, it caused a mass extinction, the second biggest of the 5 mass extinctions that we know about, if indeed we're in the 6th one now. 80% of aquatic organisms died out, and most of the plants on the land. So the fungi, extremely important in reconfiguring life, played about a bit, and the result were the forests of the Devonian, tall, arborescent lycophytes, big fleshy plants. There wasn't any lignin around at the time, just big fleshy plants. They were so successful that they also took so much carbon dioxide out of the atmosphere that they led to another mass extinction at the end of the Devonian period. So the fungi reconfigured in conjunction with the other microorganisms, and we had the Carboniferous forests, these glorious Carboniferous forests of progymnosperms and ferns, pteridophytes. And they were going the same way, carbon dioxide being dragged out of the atmosphere. And they were heading towards another mass extinction when there was some kind of biological powwow in the soils. And the fungi and the bacteria said, listen, we're trying to regulate homeostasis on this planet so that we can create human beings, etc., and everyone's happy. But we can't keep doing this boom and bust, this carbon coming out and mass extinction. How can we fix this? And the fungi said, well, do you know what? I create organic acids, don't I? Why don't I just modify some of those organic acids and create a fungi that someone one day is going to call saprotrophic fungi? What's that going to do? Well, it means that when something dies, I can recycle the carbon. Let's call it white rot fungi. They won't understand saprotrophic. Let's call it white rot fungi. And then I can put the carbon back into the atmosphere. Okay, so what have we got at the moment then in the atmosphere? Well, there's about 300 parts per million carbon dioxide. It was about 4 to 6,000 when land plants first went onto land, 5 to 6,000 parts per million. Okay, so today it's 300 parts per million. Okay, and the rest is in the ground. Yeah, all that wood and all that carbon and pollen spores in the ground rotting away. Okay, no one's going to be stupid enough to get that back out of the ground, are they? No. Okay, so we'll set life at about 300 parts per million. And give or take the odd volcanic eruption, that's how all of life has evolved over the last 300 million years. Saprotrophic fungi evolved about 300 million years ago. Let's just talk the next 3 slides, some of the things that plants do, a little bit more about horizontal gene transfer. So these are exudates. I imagine most of us know that plants secrete chemical compounds, a list as long as your arm. Some of the chemical compounds they create and pump into the soil together with the carbon sugars that feed the microorganisms are extraordinary. They'll exude flavonoids to attract bacteria, specific flavonoids, hundreds of flavonoids for very specific bacterial groups. They will exude substances called strigolactones, probably thousands of strigolactones, to attract mycorrhizal fungi. Well, the machinery, the genes that produce those flavonoids and strigolactones were given to them, were given to plants by horizontal gene transfer from bacteria about 480 million years ago. Endophytes. Not only did these microorganisms create the plants, like factories to help planetary homeostasis, but they jumped on board. This slide is taken from some research done by Professor James White at Ohio University. He was looking for into the effects of ethylene on plant stress. When plants get stressed, they give out ethylene. So he produced some stains, aniline blue, looked at plant roots, and found all these little dots. And he found concentrations of ethylene around these plant roots, and I was curious. So we looked a little bit further. This work is about 5 or 6 years ago. And he found that in the root hairs, there were bacteria. Just taking his research a little bit further, what he found was when the plants were sending out these exudates, around the meristem, where the root is just growing, there's a kind of permeability just behind the root tip. And the bacteria are being drawn in to this space, and they're being hit by a plant defense mechanism. They get hit by a substance called superoxide. And he found that that superoxide basically broke down the cell membrane of those bacteriums that had become endophytes, working inside a plant, not just in the rhizosphere, but actually inside the plant. They broke down those cell membranes, liberating the phosphorus and the carbon and the nitrogen and every other mineral in those membranes. And that was transported and was being used to feed the plant internally. Now, these little naked bacteriums, some were killed, but some didn't. Some survived. These little naked bacteriums expressed ethylene, and ethylene can be used as a growth hormone. And that ethylene was causing little root hairs to grow. And the little naked amoebas went down those little root hairs, back into the soil with a healthy dose of amnesia to reform their cell membranes and go back down. Oh, there's sugar down by the root. Excellent. Back in again. I remember this. Zapped by superoxide. And he termed this the rhizophagy cycle. And he's estimated that this rhizophagy cycle might produce up to half of a plant's nitrogen requirements. Astonishing. This little video here, if you go on to, if you look at the rhizophagy cycle on Google, you'll see these actually in video format. These are the naked bacteriums being pumped out of the root tips. Endophytes are found in every compartment, bacterial endophytes, found in every compartment of every plant. They are in the stamens, the leaves, the petals, the stems. They're like factory workers. We build a factory for a particular purpose. What do? We fill it full of offices for us to go in and work. That's what these guys are doing for planetary homeostasis. Just another little random slide. Don't have to worry too much about the writing on the left-hand side. It gives you a general kind of tick list of who's in the soil in terms of endophytes. I meant to mention as well, actually, Merlin, 80% of land plants, at least 80%, have mycorrhizal associations. So inside all of these compartments, it's not just in the soil, it's in the plants, and it's on the plants. They're everywhere. It's a bit like our gut. They're everywhere. We have protozoans inside us doing a job. We have pneumonia in our lungs doing a job, keeping other things out. Horizontal gene transfer, before I get onto this, I meant to mention this. Horizontal gene transfer, another example, is that bacteria have the ability to unravel viral DNA and make it inert. They passed that skill through horizontal gene transfer onto two other organisms, fungi and nematodes. No one else, we can't do it. Nematodes are one of the planet's great cleaner packs. They came in about 1 billion years ago. Do you know what, everyone in this audience, if you look closely tonight in the mirror, you won't see them, they're too small. But you've got nematodes probably in your eyelashes. And I bet you they're under your fingernails. And they're breaking down viruses. They're keeping you healthy, just like the rest of your microbiome. The staphylococcus on everybody's skin, MRSA, yeah, well actually, you know what? If you're healthy, like the tree, the lady asked the question about Armillaria melea on a tree, the honey fungus. It's only because that tree was poorly or malnourished, and nature is pretty relentless and says, do you know what, you're not earning your place anymore. You might have reproduced, done your job. I don't get this idea. I'm rambling now, but that's good. I don't get the idea of pathogens. Pathogens are just normal part of nature with bad PR. You know, they're just there at the end of a season to break down organic matter, like saprotrophs, yeah, like Pisium, yeah, you get on your leeks. Yeah, it's just breaking down organic matter and recycling it. Sorry, I meant to say. So this is what the microorganisms are doing. The plant is calling for different consortiums of microorganisms at the seedling, vegetative, bolting, and flowering stage. At the seedling stage, these are just the relative abundances of microorganisms, of bacteria in particular. At the seedling stage, the actinobacteriums are really important. Actinobacteriums produce antibiotics. They are the bacterial kingdom, kings of antibiotics. So when a plant is young, just like babies, mother's breast milk full of antibiotics, same principle. But when you come to the bolting and flowering stage, you're going to need nitrogen. So guess what? They put out a whole new cocktail of flavonoids to attract the cyanobacteriums, kings of producing nitrogen. One more slide before we go on to what we do about all this. For me, symbiosis doesn't cut it. Arabidopsis thaliana, penny mouse ear crest, there are 189 recorded horizontal gene transfers in this plant, virtually all of them related to metabolic processes for living on land. This isn't two organisms getting together, fungi, bacteria, and mutually benefiting. They're inventing a whole new kingdom of life. And I wanted to use this opportunity, Julia, to bring a new word in, Ingeniobiosis. This is an engineered way of life. And then we came along. Here's a potato field where the soil structure has broken down, destroyed. Most farms in this, most of the agricultural land on this planet now are degraded. Less than 2%, 3% organic matter, when it should be up in the 20%. Soil erosion, I mean, you know it, you've heard of all these stories. So what do we need to do as gardeners and growers? We need to understand that soil is a habitat It's a home. These microorganisms, they work from home. This is River Cottage. Prior to the pandemic, I was doing workshops there. In between these yellow lines is a particular geological formation. It underlies the actual garden at River Cottage. That silty sand has a pH of 7.5. Up here, up the hill, is another part of the green sand formation, has a pH of 5.5, a 200-fold difference in acidity. You can see from the gorse that's growing on the hillside, it's reflecting what's underneath. The gorse isn't growing there because it likes acidic soil. It's growing there because the microorganisms that have adapted to that kind of pH and free-draining environment are there. And then they're feeding the plant groups that prefer those environments. If we come to Colchester campus, quick look at the geology map last night. We're in the Ivor Crew building there. You'll notice that on this side, the colour of the geology is different to on that side. Everyone on this side here is sat on the Kesgrave sand and gravel formation. And roughly about here, everybody is sat on the London clay. Totally different environments, totally different microbial networks under the ground. So what can we do? So we need to understand that it is a habitat, it's a home. Now when you're in a hole, stop digging. These are 2 trial pits I did a couple of years ago next to the River Thames in Berkshire. This field hammered year after year for decades with chemical fertilizers, pesticides, herbicides, and the soil profile has completely broken down, completely compacted. 50 yards away, in a meadow that hasn't been touched for over 200 years, produces the most bountiful hay crop every year. Look at the beautiful prisms in the soil, allowing water to come up from the water table. The farmer here has to water constantly. Couldn't believe that there was water just 2 meters down. But the water can't get up to the roots, rainfall can't get down to soak the ground, but in the meadow, absolutely beautiful soil structure and abundant vegetative growth. This is my garden at home. Weeds are worthwhile. What else can we do as gardeners to improve this habitat? Leave the weeds in, never leave bare soil. All these plants are pumping carbon sugars into the ground. They're shading the ground. Not only that, but the soil is respiring. Yeah, they're breathing their aerobic microorganisms. They're breathing out carbon dioxide, precious carbon. How do they capture? How does Mother Nature want to capture carbon in bare soil? She puts plants over it. All those plants are soaking up that carbon and recycling it and putting it back into the ground as carbon sugars. Weeds are worthwhile. Never leave bare soil. Avoid plant dependency, synthetic fertilizers. This is the field I worked on in spring. There was an area here going to be used for development. So although the farmer had hedged his bets and had planted winter barley, spring barley rather, notice that he only bothered to spray that half of the field with nitrogen fertilizers. And he left this to its own devices. That soil has nothing but bacteria in it, no other mycorrhizal networks, no protozoans, no nematodes, and no worms, unsurprisingly, because there's nothing for them to eat. Well, look how sickly those plants look when they're on their own in a depleted soil. So, you get into this vicious circle of having to apply nitrogen fertilizers. Actually, just get the soil health right. Get the full complement of the soil food web going, and you won't need to. And how do you maintain healthy soil? Really good, diverse, biological, homemade composts. Not the kind of stuff you buy in a garden centre, that's been processed to death. It's organic matter. That's it. If it hasn't got the full complement of microorganisms in it, it's not compost, and it isn't going to cut it. This is a friend in my garden. We've just mixed up some biologically rich compost and making a slurry. and I'm constantly feeding the microorganisms. There's no point in putting a mulch down or a compost down at the end of the autumn. Yeah, this is the habitat. Yeah, they need to feed on a regular basis, and they need soluble carbon. So compost teas, compost slurries, absolutely the way to go. And then one final slide, bioregionality. Let's remember that, polar bears don't live in the Serengeti. So the kind of microorganisms that specialize in the cold, warmer east of the country won't necessarily be the same specialists in the mild, warm summers of the western half of the country. And look at the rainfall map over the last 30-odd years, 1971 to 2000 in the British Isles. Yeah, the microorganisms are different because they've adapted to the climate. And where you find those particular microorganisms, the plants will follow. Because right plant, right place. Thank you, Julia. Thank you, Joe. Brilliant. Thank you, everyone. Cheers.